Arvin is a resilient 31-month-old girl. Every day, her family maintains a rigorous clinical protocol to protect her brain from the cumulative damage of her condition. Thanks to a strict ketogenic diet and pharmacological regimen, Arvin has reached 18 months without a seizure, supported by clean EEG readings. Her functional cortical potential and intact motor neuron connections suggest the foundational hardware of her brain is preserved, despite developmental delays. These metrics confirm that the underlying neural infrastructure is sound. Arvin's brain is currently in a state of preserved potential, prepared for developmental growth if the molecular interference can be neutralized. The interference preventing that growth is a rare genetic condition known as DEE type 31. To stop it, we have to look at the microscopic root of the problem. Arvin's DNA contains a single spelling error, an intron 9 splice site mutation, labeled -8G to A. This disrupts mRNA processing, causing her cells to manufacture a toxic, malformed dynamin 1 protein containing two unwanted amino acids, seen here as the CR insertion. This creates a dominant negative effect, a form of active sabotage. The mutant protein actively binds to and shatters the healthy blue proteins, preventing the entire system from functioning. Arvin is currently approaching the most critical neuroplasticity window in human development. This is the narrow period between ages 3 and 5, when the brain's foundational wiring must be established. Because the mutant protein actively destroys the healthy ones, simply adding more healthy code will fail. To save her brain before this window closes, science must invent a way to selectively silence the mutation. That effort is currently underway at the Tim Yu Lab at Boston Children's Hospital, where researchers are developing a customized antisense oligonucleotide, or ASO. The ASO functions as a highly targeted chemical sniper. It is designed to enter Arvin's cells, locate the mutant genetic instructions, and destroy them before they can be translated into toxic proteins. The engineering challenge is extreme. As the crosshairs here indicate, the drug must ignore the healthy G nucleotide and lock entirely onto the mutant A. This precision prevents the drug from destroying her remaining healthy brain function. Preliminary lab data precision is achievable. Experimental candidates are already surpassing 50% efficiency in knocking down the toxic instructions. This ASO acts as a stabilization phase. By clearing away the molecular debris immediately, it protects the window of neuroplasticity, while more permanent genomic corrections are being engineered. This matrix illustrates the full scope of the strategy. The green ASO bridge provides the time needed to reach the final destination, the CRISPR base editing program at the Jackson Laboratory. Unlike the ASO, which requires repeated dosing, CRISPR base editing aims for a one-time genomic rewrite. It chemically corrects the DNA at its root, permanently changing the toxic A back into a healthy G. This timeline is accelerated by the FDA's plausible mechanism framework. By utilizing proven chemical delivery platforms like the one used for the drug nusinersen, researchers can navigate the regulatory path in years rather than decades. Recent mouse models provide the evidence for this approach. Data confirms that when the toxic protein is removed, neurological symptoms are reversible, leading to the clinical gains in motor and cognitive potential outlined here. Once the toxic interference is cleared, Arvin's protected networks will be free to connect and thrive, allowing her brain to begin the work of development. This integrated genomic roadmap leads to a first clinical application targeted for 2027, combining immediate stabilization with a permanent genetic correction. This strategy demonstrates the precision of modern medicine. Arvin's path proves that we can now illuminate a single flaw in the human code and engineer a custom repair for one individual.